Yeah. So, hello everyone. It's a great pleasure for me to introduce you the talk today. So, Mr. I'll try to speak your last name, but so here is Thales Papagiannakopoulos. Yeah. So I think it's a Greek name, right? So it's a little hard to pronounce. But anyway, it's a great pleasure to have you here, Thales. Thales is from the New York University, right? And he's here today to talk about his research. Okay. Thales is from the Department of Pathology. He has been working with cancer, molecular cancer, and he will talk about his models. He has lots of CRISPR models, cancer models that I think it's really interesting. He also works with metabolism, oxidative stress, right, Thales? So thank you very much for being here today. And please feel at home to do your talk.
Thanks so much for the invitation, Fernand. Yeah, I'm excited to be here. Um, Omji, yeah. And for those that can't pronounce my last name, my nickname at NYU is Papa G. So I get like official emails from the department that says, "Dear Doctor Papa G," and the funny thing is they capitalize the G, so it's almost like a rapper name. So anyway, okay, yeah. So as Fernando said, I'm in New York. This is Empire State Building here. This is in our this is NYU here. Multiple buildings, and our lab is on the 5th floor of this building here. And today I'm going to talk to you about some of the lab's work on lung cancer. And both the things that I'm going to present are unpublished and do not have to do so much about metabolism, but more about how tumors alter the immune system to grow. How do they use the immune system to grow better? So just to give you an idea sort of what the lab works on, so we're interested in sort of using mice and mouse models to study sort of the tumorigenic process, which starts, of course, with cells acquiring some mutations, which allow them to proliferate. Eventually, these tumor cells will have to vascularize. They become big, so they have to bring oxygen, nutrients. And as they progress, they also become quite heterogeneous. So these different colors represent different heterogeneous populations of cancer cells, which I'll show you later exist in tumors. And as tumor cells progress, they have to overcome a lot of sort of bottlenecks, as I call them, a lot of barriers, right? So nutrient availability, oxygen, they have to overcome oxidative stress that builds up, reactive oxygen species. So these are all things that we're interested in understanding, how tumor cells overcome them and how we can, by understanding that, target them with new therapies. So, and then, as I mentioned, another important thing about the tumors as they progress is they have a very altered immune microenvironment, which consists of different immune cells, which I'll discuss later today, such as myeloid cells and T cells. It's a very complex sort of environment and interactions that take place. And ultimately, of course, the tumor cells metastasize and to distant sites and kill patients. So we've been, the last few years, trying to understand how mutations that occur in patients, how they allow tumor cells to overcome some of these bottlenecks. And by understanding how they do it, we try to identify new therapies. But we're also interested in understanding how changes in whole body physiology, so for example, circadian rhythm disruption, so day and night cycles, how that affects this process. Also, how diets affect this process, which I'll talk towards the end of my presentation. And really, to look at all these and their importance in tumors, we use mouse models, which I'll discuss today. So we work on lung cancer, as I mentioned, because it's a major cause of death in both men and women in the U.S. and, of course, in Brazil, everywhere, really worldwide. And we study the major type of lung cancer that we mostly work on is lung adenocarcinoma, which is the major histological type. Um, so if you look at these tumors, they're histologically, in a single category, but if you look at the mutations that occur in these tumors, they're very diverse. So, and interestingly, now this is recent work, but from Memorial Sloan Kettering, but you can see these are the various mutations that occur in patients. But you can see here, these are grouped by mutations that are correlate with an unfavorable response, meaning if the patient has these mutations on this side here, they do worse as opposed to these mutations here. And the reason for this is because there's no good way to target these mutations therapeutically, right? EGFR mutations can be targeted using inhibitors or antibodies, but these mutations here cannot really be targeted. And the mutations I'm gonna mostly focus on today are these ones here, which are all the way on the upper right here, so that they're common. This is selection frequency, means how frequent they occur in patients, and they're very common and they're very correlated with very aggressive disease. So the other interesting thing about these mutations is that they're early mutations. So this is a study from Charles Swanton's group from UCL, where they took patient tumors and they sequenced them, and then they tried to deconstruct the evolution of the tumors. When do the different mutations occur? And what you can see is that mutations like just KRAS, KEAP1, and STK11, the ones I'm going to talk about today, are actually early events. And the reason that's important is because these mutations are found in the majority of the tumor cells. Okay, so most of the tumor cells in a patient that has these mutations will have, most of the tumors will have these mutations, and therefore targeting them is important. So there's two pathways I'm going to talk about today. One is the KEAP1 NRF2 pathway. So it relates to this mutation in this gene, this one, KEAP1. KEAP1 is a part of a E3 ubiquitin ligase complex, and it degrades this transcription factor called NRF2. So when cells build up oxidative stress, this interaction between NRF2 and KEAP1 is disrupted, and NRF2 becomes stable and goes to the nucleus and turns on transcription of antioxidant genes. So in lung cancer, what happens is you have mutations in this KEAP1 gene that basically disrupt its function, so loss of function mutation. So NRF2 gets constantly activated, and our lab has done a lot of work to understand how this works and why it's important for lung cancer. The other mutation, going back to, actually, I missed a slide here, I apologize. But the other thing I want to point out is the other reason why this is the case here, why these patients that have these mutations do not well is, we think is this, is because now in lung cancer, the majority of patients will get immunotherapy plus chemotherapy. This is the standard of care in the US and many other countries now. And what you can see here, the patients that have both these mutations here are the ones that do not respond as well to this therapy. It's very clear. So suggesting that this is one of the reasons why these patients do not do as well. So we need to understand why they don't respond and what it is about these two mutations that are impacting the ability of the immune system to kill them. So this is kind of the question, right? So is there something specific about these two mutations that are impacting the cancer cells, their environment, and the ability of the the immune cells to kill them? So this is the slide I was missing. So the other mutation there's two mutations, KEAP1 I mentioned, and this other one is STK11 LKB1. And this is a gene, it's a kinase that's upstream of multiple other kinases, signaling cascades. And these two recent studies showed that basically in lung cancer, mutations in this gene affect this signaling pathway here. So if you lose LKB1, you lose phosphorylation of SIK1, SIK3, and you lose phosphorylation of CRTC, which regulates Krebs signaling. Okay, so this study was performed by, before I get into what we did, is by Anastasia, who helped with the T-cell work, along with Ray, postdoc in the lab, and Ali, a graduate student, and with Sergey, who's an immunologist. Because I'm not an immunologist, I learned a lot from him by collaborating. So what we decided to do is we decided to use our mouse models and understand how these mutations affect lung tumorigenesis, right? So to do this, we use a a mouse model of KRAS 53 conditional Cree-LoxP mouse model. And the other trick that we have is this mouse model also has a conditional allele of Cas9. So when you deliver these mice are normally wild type, they're fine, there's no cancer. But when you deliver Cree recombinase with viruses into the lungs of these mice, the mice inhale the virus, the Cree recombinase will, in the lung epithelial cells, will activate KRAS, delete P53, and also activate Cas9. Okay, so this model will initiate tumorigenesis, and the trick we do is, along with delivering the Cree recombinase, we also deliver guide RNAs, so the CRISPR system, to the lungs directly, right? So, and in this experiment, what we did is, this is a little complicated table, but we generate these 4 different genotypes of mice. So mice that had control guide RNAs that target neomycin and antibiotic resistance, they don't target anything in the genome. So in that condition, those mice will be just KRAS 53 mutant, the tumors in the lung. We have this condition with LKB1 mutation, or KEAP1 mutation, or both. Okay, so we can generate all these 4 different conditions very quickly using CRISPR. We don't have to breed mice with conditional alleles for these different genes, right? So, okay, so, and then we can analyze various aspects about the tumors, which I'll show you: tumor burden, grade, histologically. And importantly, for what I'll show today, we can do various forms of immune profiling. So flow cytometry, immunofluorescence, single cell sequencing, which is, revolutionized a lot of our ability to look at the immune system, and also we can profile cytokines, chemokines. All right, so what we saw first of all is that if you mutate these if you have these 4 different conditions, what you see, if you mutate these 2 genes together, the tumors grow really fast, as you would expect. They're 2 important tumor suppressor genes. If you mutate LKB1 at this early time point, they also grow, and at this earlier time point, you don't see much difference between KEAP1 and wild type. But the important thing we wanted to do is we knew this was going to happen. We had an idea that the tumors would grow faster, but we really wanted to understand what is the effect on the immune system. So what we did is we used flow cytometry initially, using these combinations of markers for these various types of immune cells that are found in the lung. The lung is it's a barrier tissue, so it has a very complex set of immune cells. It has different types of macrophages that are found in different locations. Alveolar macrophages are in the alveoli. They float around basically in mucus and air, and they maintain the alveoli, versus the interstitial macrophages are actually in the lung parenchyma. And what we find is that in these conditions, when you knock out KEAP1 or LKB1, these tumors, we see dramatic depletion of alveolar macrophages and an increase in interstitial macrophages, which is very distinct. This does not happen normally in these tumors, only if you have mutations in KEAP1 and LKB1. And the other thing that happens that's exciting is we see this increase in granulocytes. These are myeloid cells that come to the lung. So we can actually look at this by single cell sequencing to get a more detailed idea of what the cells are, how they look like transcriptionally. So these are the neutrophils. It looks like this bird, this hummingbird kind of, the way this structure looks like here. But you can see wild type tumors have some neutrophils, but if you now knock out LKB1, this is LKB1 driven mostly, you can see that there's a dramatic increase in different populations of neutrophils. We try to understand now what this means, what's different about, say, this population here as compared to this one. But one thing we do know is that these purple ones here are high expression of this marker called cyclic F, which has been associated with neutrophils that are highly immunosuppressive, that suppress T cell function. The other population we looked at are macrophages, and they're even more complex. There's a lot of different macrophages in the lung. So you can see here that different colors represent different populations that we find, and there's a lot of them. This is the control condition, and you can see there's quite dramatic differences when you go to the different mutations. And one of the major things that happens is you see a decrease in this blue cloud here. These are the alveolar macrophages. They almost disappear. You can see the quantification here. And the other thing that happens is you see an increase in macrophages down here, these populations, which are really high in this marker called arginase 1, which is an immunosuppressive marker. Arginase 1 is an enzyme that degrades arginine in the microenvironment, and by doing so, it suppresses T cell function. But you can see how having specific mutations really changes the function or the transcriptional state of the immune cells, not just their numbers, but their transcription. And we can visualize it. So this is cool, but maybe this is computational. This is all computational. Maybe it's not real cells that exist in the tumors, but we can actually visualize them using microscopy. So you can see here, this is a control tumor. Looks kind of boring, If you look at the tumors here, you can see there's Ly6G is neutrophils. You can see there's a lot of neutrophils in these tumors. Arginase 1 in orange, you can see there's a lot of it. And it co-localizes with F480, which is like a macrophage marker. So the idea is that we can actually show now that these populations exist in the tumors, and we want to understand, how do they get there and what do they do. So the other important thing is that we can actually see these in human tumors. So this is we got this. It's a tumor microarray. So you have like cores from different patients, and each dot here represents a different patient. So we have quite a few, and we know the mutations in these patients. These are the mutations. And we can actually here stain for MPO, which is a neutrophil marker. And as I showed you before, in mouse tumors, LKB1 mutant tumors have high neutrophil infiltration. You can see it here by quantification. You can also visualize here in brown staining. These are neutrophils. So what we see in mouse is true in humans too, which is important because we want to be able to study biology that's relevant in patients as well. So what we've seen so far, and I'm simplifying some of the data, but I don't have time to show you all the profiling that we've done. But the idea is that when you have these mutations, we see high interstitial macrophages, neutrophils, and a decrease in alveolar macrophages, right? So, we have two questions, right? One question is, how does this happen? How do these change? Is it because of the mutations in the tumor? And how does that impact the T cell function, right? The cells are supposed to come and kill the tumor. So to do this, to understand this, what we did is we took the tumor cells, we sorted them out, and we did RNA sequencing. We also did, so that's this here. We also did, collected, so you can collect bronchoalveolar lavage, so you can before sacrificing mice, you can inject PBS into their lungs through the trachea, collect the fluid back, and use that to profile for cytokines, like protein levels, using ELISAs or Luminex assays. And we also looked at tumor cell condition media in vitro. So, and we compared. And what we found is that there was these major cytokines here that were changing. So some of these are actually, you probably know about IL-6, CSF3 also is a known neutrophil chemoattractant, 601, 5, IL-33 was already somewhat known, but the one that caught our attention was this one here, LIF, because this was not previously known to be important in lung cancer at all. And it's mostly known for its role in stem cell biology. It plays a role in stem cell differentiation and renewal. So LIF is, what is LIF? It's a cytokine. It binds to its receptor. That's a heterodimer of LIF receptor and also GP130, which is also part of the IL-6 receptor. So so LIF is an IL-6 family member. And when it does this, when it binds, it stimulates primarily STAT3 signaling through the JAK-STAT signaling pathway. And this can play a role, as I mentioned, in survival, self-renewal, differentiation. However, it was really not understood if it plays a role in cancer, in lung cancer, and potentially for immune responses. So first of all, just to show you, it's very dramatic. What you see is in healthy lungs from mice, there's no LIF. In KRAS53 mutant tumors alone, no LIF. But now if you mutate LKB1 and keep one together, you see a very strong increase in LIF. we did in situ hybridization, and you can see this is the only way we can detect it by microscopy. And you can see that we can detect its expression in the tumors. There's nothing in the wild-type tumors. One thing to point out is it's heterogeneous. It's not everywhere, so that's interesting. Not all the tumor cells express LIF, but you can see that it's high in the tumor cells by expression, and also in the lung. It's secreted. This is by ELISA. So what we thought may be happening is that maybe LIF signaling, LIF is made by the tumor cells and signals to the tumor cells through the STAT3 signaling to promote cytokine production. This is the hypothesis. So to test this, first thing what we did is we checked and we asked, do the tumor cells with these mutations have more STAT3 signaling? And in fact, that's what we saw. We see the same thing in human tumors with these mutations. So this is not just in mouse, it's also true in humans. So that's great. So then we wanted to ask the question, is LIF made by the tumor cells and signaling to the tumor cells to promote these cytokines, right? So how do we test this? We do two things. We did genetics first. So again, this table is a little complicated, but what we did is we used our system here that I showed you before, this viral system, where we have two different promoters, and one promoter has expresses this guide RNA, and another one these guide RNAs, right? So in this guide position, we put always LKB1, so we muted LKB1. And in the other guide position, we put a control. We knock out LIF itself or its receptor, okay? So by doing this now, we generate LKB1 or LKB1 keep-on mutant tumors. We've done both. And now we have a control condition, a condition we delete LIF or delete LIF receptor only in the tumor cells, okay? And we do our profiling and our various downstream analysis, right? So what we saw, which was exciting to us, was when you knock out LIF, this is MRI, or LIF receptor, now you can see that it has the same effect. The tumors basically decreased in size dramatically, and this is quantified here. And this is only in the tumor cells, okay? So this suggests that LIF that's made by the tumor cells binds to LIF receptor on the tumor cells to do autocrine signaling. And the other reason we think this is true is this is measuring LIF by ELISA in the bronchoalveolar lavage, in the fluid in the lungs. And you can see when you knock out LIF in the tumor cells, it almost completely goes away, suggesting that most of it does come from the tumor cells, so that's good. And this is also really cool here is what you see is when you knock out LIF receptor, the tumor cells are almost, decreased dramatically, their size. However, LIF is still there, but it's not signaling, right? There's no LIF receptor, so even though it's there, the tumors cannot process it. They cannot bind to it. So the other thing we wanted to do is we wanted to know, is this therapeutically relevant? So what we did is this is a very expensive experiment. we had to make our own, generate our own LIF antibody, and that was, it was very costly. But anyway, we did it. We had to do it. So what we did is we initiate tumors. In this case, just LKB1 mutant. We let the tumors grow for 8 weeks to establish, and then we used we injected the mice with an antibody that neutralizes LIF, so binds to it and blocks its ability to signal, right? So, and we did this for 4 weeks, and we looked at what happens to tumor burden, and what we saw, which was exciting, was not just genetically, but now using antibodies, we can actually block LIF function and dramatically suppress tumorigenesis. It was a very robust effect. The other cool thing was that we saw when we look at other cytokines in the lungs now, we saw that they're all LIF dependent. So you can see here, when you block LIF, LIF itself goes away, so that's good. We're neutralizing it, but IL-6 goes down, CSF3, 601, CCL2, which is important for monocytes, for myeloid monocyte cells to come to the lung. So suggesting that this could be a new therapy, targeting LIF, a new therapy in the clinic to target tumors with LKB1 keep-on mutations, and potentially to promote immune responses, which I'll show you in a minute. And the other thing that we saw is, as you would expect, if we knock out LIF or LIF receptor, STAT3 goes down in the tumor, so that's good. So again, it's autocrine signaling. So Some cool stuff that I wanna show you that I was kind of excited because I like immunology. It's interesting, but also I'm a tumor biologist. So is this? So we did single cell sequencing um, in the tumor cells. So these are just tumor cells here. These are the 3 conditions. Okay. We detected 8 different clusters, the different colors here. Okay. So these are different transcriptional states that exist in the tumors, right? Different populations. So one thing I want to point, 2 things I want to point out is that these 2 populations here decrease quite a lot or almost completely in this case, when you knock out LIF or LIF receptor. So these tumor cells just go away, especially these ones here. And what's important about these tumor cells is this was quite surprising to us, was that these are the ones that are primarily making, this is by single cell sequencing, are making the majority of the cytokines. So the tumor cell populations that are making the majority of the inflammatory cytokines, are the ones that disappear when you lose LIF receptor. They go away. And this is another way to show it, using transcriptional signatures of inflammatory response. In blue, these are cells that have high signature, and you can see these populations have high, and they're gone completely here when you knock out LIF receptor. Okay, so the other thing that's really interesting about these 2 populations here is that they are the ones. So NKX 2.1 is every cell in the lung that's a lung epithelial cell expresses NKX 2.1. Without NKX, if you make mice that are NKX 2.1 knockouts, they can develop until the time they have to make lungs, and then they cannot make lungs without NKX 2.1. They die prematurely. So it's an important transcription factor for lung identity. So what happens is, as the tumor cells, as the tumors grow, some populations of the tumor start losing NKX 2.1, as you can see here. And these are the populations of the tumor cells that are more dedifferentiated, like a sort of EMT, epithelial mesenchymal kind of state. And what's interesting is that these are the ones that actually disappear with LIF signaling loss. Okay, so, and what's also kind of interesting we're trying to study now is that even though they start expressing markers that are not normally expressed in lung epithelial cells, such as SOC17. SOC17 actually in normal lung is only expressed in endothelial cells in blood vessels. So they have this weird identity. So, and we actually find these cells in human tumors too, in small populations. So that we think they're actually real. But they're important because they are the ones that are actually, we think, making tumors more immunosuppressive. They make cytokines that recruit immune cells, and by doing so, they generate an immunosuppressive environment. It's not, we don't think it's all the tumor cells that are doing this. We think it's specifically these populations here. So, and just to show you that, we also looked at, okay, so I showed you LIF signaling is important for tumor growth. I showed you its specific populations that are making the cytokines. When we block it, the inflammatory cytokines go down. But what happens to the immune cells? Do they change? Yes, and this is just the easiest example to show here. You can see if you knock out LIF or LIF receptor, basically arginase 1 macrophages almost disappear. And we see the same thing when we use an antibody against LIF. So, and you can see visually here, arginase is basically gone in these tumors by fluorescence. Ly6, this is the blue color. This is Ly6G, which is neutrophils. They're gone. So, so we really, LIF signaling is very important for maintaining these myeloid cells in the tumors. So this is sort of the model. We think that basically tumor cells secrete LIF. It signals to the tumor cells themselves. And by doing so, it promotes cytokine production, chemokine that recruits these and differentiates these immune cells, these innate immune cells, to immunosuppressive states. So, but now the question is, if that's true, what happens to the T cells, right? I didn't show you any T cell data. So, so what we see is if you knock out LIF or LIF receptor, you see an increase in interferon gamma producing CD4 and CD8 T cells. TNF alpha also goes up. Sorry. I'm just gonna skip here. And the other thing that we see is that if you do single cell sequence, the same single cell data that I showed you before, you look at the T cells. These are the different T cells that we detect. There's CD4 T cells here, CD8 T cells here. And in this case, what we do is we can actually sequence the T cell receptor. So we know the sequence of the T cell receptor, which is indicated. So, and T cells that have the same T cell receptor sequence means that they're actually clonally related. So it means they were generated from a single T cell that proliferated. And it's a little complicated sometimes to explain this, but the different dots here represent different T cell receptor sequences. And the colored dots here are the ones that where you have multiple T cells with the same receptor. And you can see that in the control, you can't detect them. But if you look at LIF or LIF receptor, now you an expansion of these populations here, suggesting that these T cells are coming into the tumor and proliferating and detecting a tumor antigen, right? That's the important thing. And we can show that they're functional because if you take now these mice here, the tumors, as I showed you, do not grow well. If we treat these mice for 8 weeks after tumor initiation with CD4, CD8 antibodies to deplete the T cells, get rid of the T cells, you can see now that the tumors can grow again, suggesting that in this condition here, the T cells are suppressing tumor growth, okay, and which is LIF dependent. Okay, so this is the model again, looking at tumor cells per se. You can see that we've shown that, and I didn't show you all the data, but we know that LKB1 through the signaling axis regulates LIF, which then binds to its receptor on the tumor cells, regulates STAT3 signaling, which promotes the production of a lot of pro-inflammatory cytokines. This is important for tumor growth, and we think we can target this now using neutralizing antibodies. So hopefully this can be an approach in the clinic. I didn't really talk about what KEAP1 does here, and I don't have time for this today, but we think KEAP1 plays a role mostly in regulating T cells directly through metabolic regulation, and we have a paper in bioRxiv that touches on this. We're still working on this in the lab. And there's a lot of things we need to understand still. Like, for example, some of these cytokines, we don't really know exactly what their role is in regulating the immune response. We know that they're downstream of LIF, but we don't know what they're actually doing. We don't know how heterogeneous the LIF signaling is. So as I showed you before, very few tumors, specific tumor cells make LIF. It's not all of them. And actually, we call it autocrine, but it could be that some tumor cells are making LIF and it's signaling to other tumor cells. It's not the same cell exactly. So we have no idea. So that's we're looking into this with single cell data now. So this is the first part of the talk. And because Fernando told me you guys are interested in nutrition. Oh, yes. Maybe we can pause it if that's okay. Okay. It's okay.